So today I was able to speak about innate immunity and its role in the pathogenesis of MDS. And what we've learned is that there's a very important cellular effector of innate immunity is what we call myeloid-derived suppressor cells, or MDSCs, and they're dramatically expanded in MDS. One of the reasons for that is a profound upregulation of an inflammatory protein called S100A9 that drives the expansion of MDSCs, which actually suppress blood development and cause injury to those cells. What we've learned is that innate immune activation, which is driven through the toll-like receptor families, drives not only the expansion of the MDSCs, but it also activates another signaling pathway in innate immunity called the inflammasome. And the inflammasome is a signaling complex that will, when it's activated, it'll drive not only proliferation of the cells, it'll also generate inflammatory proteins such as interleukin-1, but it also can then cause a specific type of cell death called pyrotosis. And pyrotosis is a lytic type of cell death, so it'll release all the intracellular contents, and those intracellular contents are noxious stimuli for toll receptors, so it continues to reinforce that process. I think that there's a number of things that we can do for targets to intervene. One of the things we've also learned is this pathway can be activated not just from cell extrinsic factors like S100A9 or other toll-like receptor ligands, but also from cell intrinsic effects, such as genetic mutations. They all license the same pathway called the inflammasome. So things that inhibit the inflammasome, and there are some of those agents in development, inflammasome inhibitors, probably getting into clinical trials later this year. That would be one good strategy to pursue. The other is try to neutralize some of the ligands that are circulating that actually activate this pathway. So one of the first ones being studied is canakinumab from Novartis. That's an antibody, a humanized monoclonal antibody that'll neutralize interleukin-1, which is part of the toll-like interleukin receptor family, which can activate this pathway. So that's being studied in patients who have failed recombinant erythropoietin, and the endpoint of that is primarily to improve red blood cell development. There's other strategies, whether it's neutralizing S100A9 or the signaling pathway through the toll-like receptors that we call the myddosome. There are now agents that inhibit IRAK1, IRAK4, or the combination. Those are the key kinases downstream from the toll-like receptors.